السلام عليكم أعزائي الطلبة، وأرحب بكم بالعام الدراسي الجديد. العام الدراسي الجديد سيكون التدريس بأسلوب محدث. ستكون هنالك محاضرات أونلاين، وسيكون هنالك المحاضرات العملية يكون حضوري، ويسمى هذا التعليم المدمج. سنتكلم خلال الكورس الأول على الجهاز المناعي لجسم الإنسان، وسيكون المحاضرة هي عبارة عن overview of the immune system. We will start with the lecture of the innate immune response, or we call it non-specific immune response. What is the teaching objectives? First, to recognize the significance of the immune system. Second, to distinguish between the innate and the adaptive immune system. Third, to understand the mechanisms of combating infection or disease to kill the pathogen. Fourth, to know the humoral and cellular component of the immune response. Fifth, to recognize the mechanism of action of the component of the innate immune response. An overview of the immune system. We are constantly being exposed to infectious agents, and yet, in most cases, we are able to resist these infections. It is our immune system that enables us to resist infection. The immune system is composed of two major subdivisions: the innate and adaptive immune system. Also, we call the innate, we call it the non-specific immune response, and the adaptive, we call the specific immune system. The innate immune system is our first line defense against invading organism, while the adaptive immune system acts as a second line of defense and also afford protection against re-exposure to the same pathogen. Each of the major subdivisions of the immune system has both cellular and humoral component, by which they carry out their protective functions. In addition, the innate immune system also has an anatomical feature that functions as a barrier to infection. Although these two arms of immune system have distinct function, there are interplay between these systems. The component of the innate immune system influence the adaptive immune system and vice versa. Here we talk about the protection against infection. Our body is designed to protect against invading organism. As we see, our anatomical barrier is the first line defense mechanism, like skin, mucous membrane, and the certain secretions that present in our gastrointestinal systems. All these systems protect the body against invading organism. Collectively, these systems we call the innate immune systems. After breaking the immune system and entrance of pathogen to our body, the second line immune systems will defense to protect the body, and this is we call the adaptive immune systems, and we will talk it in details in the subsequent lectures. There are two phases of the immune response: pathogen recognition and pathogen removal. Although the innate and adaptive immune system both function to protect against invading organisms, they differ in a number of ways. The adaptive immune system requires some time to react to an invading organism, whereas the innate immune system includes defense that mostly are constitutively present and ready to be mobilized upon infection. Second, the adaptive immune system is antigen specific and reacts only with the organism that induced the response. In contrast, the innate immune system is not antigen specific and react equally well to a variety of organisms. Finally, the adaptive immune system demonstrates immunological memory. It remembers that it has encountered an invading organism and react more rapidly on subsequent exposure to the same pathogen. In contrast, the innate immune system does not demonstrate immunological memory. So, the role of immune system is to recognize a foreign antigen that may be harmful to the body and to remove the invading microorganism. أعطيكم مثال بسيط. اليوم أثناء جولتي داخل الكلية رأيت بعض الطلبة والطالبات مع الأسف ما ملتزمين بالوقاية، ما لابسين كمامات، فاضطريت أرجع إلى الاستعلامات مع الكلية وبلغت بأنه يمنع دخول الطلبة اللي ما ملتزمين بشروط الوقاية الصحية. فموظف الاستعلامات بدأ بتوجيه الطلبة بلبس الكمامات. بعد فترة نزلت مرة ثانية للساحة، وجدت بعض الطلاب ما ملتزمين بارتداء الكمامات. إذا أنا شخصتهم بأن هؤلاء هم باثوجين. زين، العمل معهم شو يكون؟ توجيههم بأنه لبس الكمامات أو باثوجين ريموفال. فنبهتهم بأنه يطلعون خارج الكلية. هو الغاية من هالموضوع بأنه نقول لهم يا طلابنا يا أعزائنا، إحنا دا نشخصكم أنتم ما ملتزمين. فنتيجة عدم الالتزام لازم يكون ردة فعل من الكلية بمنعهم دخول حرم الكلية، وذلك خشية على بقية الطلبة الملتزمين في شروط الأمان. الآن سنتكلم على cells that constitute the component of the immune system. All cells of the immune system have their origin in the bone marrow. They include myeloid cell line like neutrophils, basophils, eosinophils, macrophage, and dendritic cells, and lymphoid cell line like B lymphocyte, T lymphocyte, and natural killer cells. This figure shows the cells that involved in the immune system, where there is two major precursor cells of the immune system, which is the myeloid cell line that include the granulocyte and monocyte, and the lymphoid cell line that will include the T, B, and natural killer cells and their ancestors. Now, what is the function of the immune system? The main function of the immune system is cell to non-cell discrimination. The ability to distinguish between self and non-self is necessarily to protect the organism from invading pathogen or microorganism, and to eliminate modified or altered cells like malignant cells. Since pathogens may replicate intracellularly, like viruses, some bacteria and parasites, or extracellularly, like bacteria, fungi, and parasites, different components of the immune system have evolved to protect against these different types of organisms. It is important to remember that the infection with an organism does not necessarily mean disease. يعني مو شرط بأنه ال entrance of microorganisms that cause disease. Why? Since most cases the immune system will be able to eliminate the infection or microorganism before disease occurrence. Disease occurs only when the bolus of the infection is high or when the virulence of the invading organism is great or when the immunity is compromised. هي موازنة بين شدة الإصابة بالميكروبس وبين المناعات الجسم. Although the immune system for the most part has beneficial effect, there can be detrimental effect as well. During inflammation, which is the response of the invading organism, there may be local discomfort and collateral damage to healthy tissue as a result of toxic product produced by the immune response. In addition, in some cases, the immune response can be directed toward self tissue and resulting in autoimmune disease. After entrance of the organism, there will be an inflammatory process, cell recruitment, fluid collection, fever, and so on. All these processes may be harmful to the body, but at the same time, it to protect the body from invading organism. Now we will start with the immune system and the beginning with the innate defenses against infection. It will be differentiated into different categories. One of them is the mechanical or the anatomical barrier. The anatomical barrier will be classified into mechanical factors, chemical factors, and biological factors. Mechanical factors, epithelial surface form a physical barrier that is very impermeable to most infectious agents. Skin. Thus, the skin acts as our first line of defense against invading microorganisms. The desquamation of the skin epithelium also helps remove bacteria and other infectious agents that have adhered to the epithelial surfaces. Movement due to cilia or peristalsis help to keep air passage and the gastrointestinal tract free from microorganisms. The flushing action of tears and saliva helps to prevent infection of the eyes, mouth, and trapping effect of the mucus that line the respiratory and gastrointestinal tract help to protect the lungs and the digestive system from infection. The hair that grow in the nostrils and the skins as part of the mechanical barrier. The second barrier is the chemical barrier. Fatty acids in sweat inhibit the growth of bacteria. Lysozyme and phospholipase found in tears and saliva and nasal secretions can break down the cell wall of bacteria and destabilize the bacterial membrane and result in cell death. The change in the pH of the sweat and gastric secretions prevent growth of bacteria, like high acidity of the acids in the stomach. Defensin, which is a low molecular weight protein found in the lung and gastrointestinal tract, have antimicrobial activity. Surfactant in the lung act as an opsonine. We will talk it later on. The third one is the biological factor. The normal flora of the skin and gastrointestinal tract can prevent the colonization of pathogenic bacteria by secreting toxic substance or by competing with pathogenic bacteria for nutrient or attachment to the cell surface. The other part of the innate system is the humoral barrier. Anatomical barriers are very effective in preventing colonization of tissue by microorganisms. However, when there is damage to the tissue, the anatomical barrier are breached and infection may occur. جرح داخل الجلد. إذا the inner structure will be exposed والmicroorganism will enter and cause inflammation. Once infectious agents have penetrated tissue, another innate defense mechanism come into play, namely acute inflammation. Humoral factors play an important role in the inflammation, which is characterized by edema and the recruitment of the phagocytic cells. These humoral factors are found in the serum or they are formed at the site of infection. The other part is the complement system, is the major humoral non-specific defense mechanism. Once activated, complement can lead to increased vascular permeability, recruitment of phagocytic cells, and lysis of the opsonization of the bacteria. Depending on the severity of the tissue injury, the coagulation system may or may not be activated. Some product of the coagulation system can be contribute to the non-specific defense because of their ability to increase vascular permeability and act as a chemotactic agent for phagocytic cells. In addition, some of the products of the coagulation system are directly antimicrobial agents. For example, beta-lysine, which is a protein produced by platelet during coagulation, can lyse many gram-positive bacteria by acting as a cationic detergent. By binding iron is an essential nutrient for bacteria. Lactoferrin and transferrin limit bacterial growth. Lysozyme break down the cell wall of bacteria. Cytokines have a various effect depending on the balance. Interferon or protein can limit virus replication in cells. Some interleukins induce fever and the production of acute phase protein like C-reactive protein, some of which are antimicrobial because then they can opsonize the bacteria. The other part of the innate immune system is the cellular barrier. Part of the inflammatory response is the recruitment of neutrophils, eosinophils, and macrophage to the site of infection. These cells are the main line of defense in the non-specific immune system, the innate immune system. Neutrophils are recruited to the site of infection where they phagocytize invading organism and kill them intracellularly by engulfment. In addition, neutrophils contribute to collateral tissue damage that occurs during inflammation. tissue macrophage and a newly recruited monocyte, which differentiate into macrophage after migration from the inside the blood vessels, also functions in phagocytosis and intracellular killing, like like neutrophils. In addition, macrophages are capable of extracellular killing of infecting or altered self-target cells. Furthermore, macrophages contribute to tissue repair and act as an antigen-presenting cells, which are required for the induction of a specific immune response. It is the link between the innate and the adaptive immune response. The other one is the natural killer cells and lymphokine activated cells can non-specifically kill viral infected and tumor cells. These cells are not part of the inflammatory response, but they are important in non-specific immunity to viral infection and tumor surveillance. Another cell is the eosinophil, have a protein in their granules that are effective in killing certain parasites. الآن نتكلم على تفاصيل أكثر حول phagocyte response to infection. Circulating neutrophils and monocytes respond to danger signals generated at the site of infection. These signals include N-formyl methionine containing peptides released by the invading bacteria, clotting system peptides, complement product, and cytokines released from tissue macrophages that have encountered bacteria in tissue. Some of the SOS signals stimulate endothelial cells near the site of infection to express cell adhesion molecules like ICAM1 and selectins, which bind to the component of the surface of macrophage and cause the phagocyte to adhere to the endothelium. Vasodilator produced at the site of infection cause the junction between the endothelial cells to loosen and the phagocytes then cross the endothelial barrier by squeezing between the endothelial cells in a process called diabetes. Once the tissue spaces, some of the SOS signals attract phagocyte to the infection site by chemotaxis. The SOS signals have also activate phagocyte, which result in increased phagocytosis and intracellular killing of the invading microorganism. So this the site of an inflammatory reaction when there is vasodilatation and increase spaces between the endothelial cells, the phagocytic cells will migrate into the tissue by a process we call the diabetes. After that, it will be stimulated by signals from the product of the phagocytic cells itself to activate their own system, and certain chemo attractant substance will cause more migration of the phagocytic cells to the site of inflammation and will result in killing of the. Now we'll talk on some details of phagocytosis and killing of the microorganism. This process called phagocytosis. Phagocytic cells have a variety of receptors on their cell membrane through which infectious agents bind to the cells. These include Fc receptors, complement receptors, scavenger receptors, and toll-like receptors. After attachment of the bacterium to the to the receptors, the phagocyte began to extend their pseudopods around the bacterium. The pseudopods eventually surround the bacterium and engulf it, and the bacterium is enclosed in the phagosome. During phagocytosis, the granules or lysosome of the phagocyte fuse with the phagosome and empty their content. The result is the bacterium engulfed in a phagolysosome, which contain the content of the granules or lysosomes. These content will participate in the killing of the bacterium. So this is the phagocytic cells, and this is the bacterium attached to specific site of receptor on the phagocytic cells. After that, there will be an engulfment of the bacterium. Then the engulfed bacteria within the lysosomes, these will result in release of certain enzymes and antioxidants that will result in killing of the bacteria. Now we will talk about the process of phagocytosis and how the phagocytic cells like macrophage and neutrophils kill the invading bacteria or other microorganisms. The phagocytic cells have a variety of receptors on their cell membrane through which the infectious agent like bacteria or viruses bind to the cells. These include the Fc receptors, complement receptors, scavenger receptors, and toll-like receptors. After attachment of the bacterium, the phagocyte begin to extend their pseudopods around the bacterium, like in figure B. The pseudopods eventually surround the bacterium and engulf it, and the bacterium is enclosed in a phagosome. During phagocytosis, the granules or lysosome of the phagocytic cells fuse with the phagosome and empty their content. The result is the bacterium engulfed in a phagolysosome, which contain the content of the granules or lysosomes. These figures show the steps in the engulfment of the bacterium with the subsequent killing them. Now we will talk on some details on phagocytosis and engulfment of the bacterium. Phagocytic cells have a variety of receptors on their cell membrane through which infectious agents bind to the cells, like Fc receptors, complement receptors, scavenger receptor, and toll-like receptor. After attachment of bacterium, the phagocytes begin to extend the pseudopods around the bacterium. The pseudopods eventually surround the bacterium and engulf it, and the bacterium is enclosed in a phagosome. During phagocytosis, the granules of or lysosome of the phagocytes fuse with the phagosome and empty their content. The result is the bacterium engulfed in a phagolysosome, which contain the content of the granules or lysosomes. These processes result in killing and degradation of the bacterium. This figure shows the process of attachment, penetration, and entrance of the bacterium, and the result in killing of the bacteria. Now we will go deeper on the mechanism of killing of engulfed bacteria by phagocytic cells. Respiratory burst. During phagocytosis, there is an increase in glucose and oxygen consumption, which is referred as the respiratory burst. The consequence of the respiratory burst is that a number of oxygen-containing compounds are produced, which kills the bacteria being phagocytized. This is referred to as oxygen-dependent intracellular killing. In addition, bacteria can be killed by pre-formed substance released from granules or lysosome when they fuse with the phagosome, and this is referred to as oxygen-independent intracellular killing. Oxygen-dependent myeloperoxidase independent intracellular killing. During phagocytosis, glucose is metabolized via the pentose phosphate shunt and NADPH is formed. Cytochrome B, which is part of the granules, combined with the plasma membrane, NADPH oxidase and activate it. The activated NADPH oxidase uses the oxygen to oxidize the NADPH. The result is the production of superoxide anion. Some of the superoxide anion is converted into hydrogen peroxide and singlet oxygen by superoxide dismutase. In addition, superoxide anion can react with the hydrogen peroxide, resulting in the formation of hydroxyl radical and more singlet oxygen. The result of all these reactions is the production of the toxic oxygen compound, superoxide anion, hydrogen peroxide, singlet oxygen, and hydroxyl radicals. All these are reactive oxygen species. Oxygen-dependent myeloperoxidase dependent intracellular killing. As the azophilic granules fuse with the phagosome, myeloperoxidase is released into the phagolysosome. Myeloperoxidase utilizes hydrogen peroxide and halide ions, usually chloride, to produce hypochlorite, a highly toxic substance. Some of the hypochlorite can spontaneously break down to yield singlet oxygen. The result of these reactions is the production of toxic hypochlorite and singlet oxygens. All these are we call the reactive oxygen species. The final steps in the mechanism of phagocytosis is the detoxification reaction. Polymorph nuclear cells and macrophages have means to protect themselves from the toxic oxygen intermediates. These reactions involve the dismutation of superoxide anion to hydrogen superoxide by superoxide dismutase and the conversion of hydrogen peroxide to water by catalase. These figures summarize the steps in the generation of reactive oxygen species and different steps. Oxygen independent intracellular killing. In addition to the oxygen dependent mechanism of killing, there are also oxygen independent killing mechanisms in phagocytosis. Cationic proteins, cathepsin, released into the phagolysosome can damage bacterial membrane. Lysozyme break down bacterial cell walls. Lactoferrin chelate ions, which deprive bacteria from this required nutrient. Hydrolytic enzyme break down the bacterial protein, thus even patients who have defect in the oxygen-dependent killing pathway are able to kill bacteria. However, since the oxygen-dependent mechanisms are much more difficult in killing, patients with defect in these pathways are more susceptible to get a more serious infections. Nitrogen oxide-dependent killing. Binding of bacteria to macrophage, particularly binding via toll-like receptors, result in the production of tumor necrosis factor alpha, which act as in an autocrine manner to induce the expression of the inducible nitric oxide synthetase gene, resulting in the production of nitric oxide. If the cells is also exposed to interferon gamma, additional nitric oxide will be produced. Nitric oxide released by the cells is toxic and can kill the microorganism in the vicinity of the macrophage. These figures shows the killing of microorganism by different cytokines. Non-specific killer cells. Several different cells, including natural killer cells, activated macrophage, eosinophils, and mast cells, are capable of killing foreign and altered cell target cells in an unspecific manner. These play an important role in the innate immune system, and these will be talked later on in subsequent lectures. Innate response to virus infection, altered cell transformed cells. NK cells have two kinds of receptors on their surface: NK receptor and inhibitory receptor. When the NK receptor encounter its ligand on the target cells, the NK cells is signaled to kill. However, if the inhibitory receptor is also bind to its ligand, which is the MHC class 1, then the killing signal is repressed. Normal cells constitutively express MHC class 1 on their surface. However, viral infected and transformed cells downregulate expression of these MHC class 1 molecule. Thus, NK cells selectively kill viral infected and transformed cells while sparing normal cells. And this is depend on the expression of MHC molecule. Innate response to extracellular microorganism like parasite. Eosinophils are a specialized group of cells with the ability to engage and damage large extracellular parasites such as schistosomas. Activated eosinophils release their granules component including major basic protein, eosinophil peroxidase, cationic hemoprotein, and eosinophil cationic protein, ribonuclease, that is an eosinophil specific toxin that is very potent in killing many parasites. Determinant recognized by the innate immune response. Determinants recognized by the component of the innate immune system differ from those recognized by the adaptive immune system. Antibodies and B and T cells receptor recognize discrete determinant and demonstrate a high degree of specificity, enabling the adaptive immune response to recognize and react with a particular pathogen. In contrast, components of the innate immune system recognize a broad molecular pattern found in a pathogen that is not in the host. Thus, they lack a high degree of specificity seen in the adaptive immune system. A broad molecular pattern recognized by the innate immune system have been called PAMPs, pathogen-associated molecular pattern, and the receptor for PAMPs are called SPRRS, pattern recognition receptors. A particular PRR can recognize a molecular pattern that may be present in a number of different pathogens, enabling the receptor to recognize a variety of different pathogens. Example of some PAMPs and PRRs are illustrated in figures below.